we'll be able to reintroduce Penny Reynolds, who is going to talk to us today about preproducibility and backwards design, especially in animal research, preclinical animal research. Penny, thank you so much for joining us here today.
Okay, I apologize for the technical difficulties, but it worked out quite well because I can segue into my talk from Lisa and Miriam's more general recommendations and just focus on a specific branch of research which uses animal models. So why preclinical models? Well, no matter what you think about the use of animals in research, they're nevertheless absolutely essential for trialing various therapeutic agents or devices before we proceed to trying them out in human beings. They're important for establishing the pathobiology, the mechanisms of disease, for identifying target molecules and mechanisms of action so you can assess the therapeutic potential of the agent, and also for assessing therapeutic benefit and, of course, potential harm. So unfortunately, though, the whole clinical trial process is not particularly efficient. At least 90% or more of clinical trials will fail because they either don't work or they kill people. So failure occurs very early. I mean, it's one of the principles of process engineering and quality control, but you have to look upstream for the solution to downstream problems. And it's a unfortunate rule of thumb by industry that at least half, probably more, of academic studies cannot be repeated by industry. So the situation is probably even more dire than that because of the famous landmark replication studies are definitely symptomatic of translational problems. Fair healthcare, or roughly 1 in 5 published findings could be validated. The Amgen study of preclinical oncology studies, less than 1 in 10. Reproducibility project for cancer biology. The attempt was to replicate at least 50 high-impact studies. After massive cost and time overruns, they could only do about 18. Results very inconclusive. And then in toxicology, analyzed over 2,000 drugs in rodent models, no better than a coin flip. In a couple of years ago, Phil Stark defined the prerequisite for reproducibility as being what he called preproducibility. And this has been a point that's been emphasized by a number of other speakers, is that we need clear and complete communication of relevant methodology to allow others to replicate or verify the results. One of the problems is that if nobody does the methods in the first place, they can't be reported. If you don't do it, you can't report it. And if you don't report it, it can't be verified. So part of the problem is that a lot of investigators see study as a pipeline. They do their experiment, they collect the data, they do statistical analysis, p less than 0.05. Yes, yay, done. No, you either bin the study or go back and do it again. The problem with this model is that there's undue emphasis on statistical significance and relevant methodology is going to be overlooked. In reality, this is really what a study looks like. It's complex, it's nonlinear, it's dynamic, with a lot of moving parts. I suggest that we borrow a concept from classroom teaching and curriculum design, which is backwards design or planning in reverse order. We identify the results that are desired, we determine the evidence that would be acceptable, and we plan the study and design a protocol around that. And Wiggins and McTighe more or less categorize these prerequisites as 3 Ps. We need authenticated performance metrics, how we know it works. It has to be intentional. The purpose of each task has to be established before implementation, and planning has to be transparent, complete, and explicit. So, applied to research, what are our desired results? High quality, reproducibility. What criteria are evidence of Q&R? Well, we need acceptable metrics, and how do we build those metrics into our study design and protocol? To identify the desired results, we have to do a little bit of a deeper dive into what reproducibility is. And what it's really asking is, are the underlying data trustworthy? So reproducibility requires that the model be representative, and it also requires that the data are reliable. So what the points are I want you to get from this slide is that, first of all, reproducibility is built on a foundation of sound methodology followed by truthful, accurate reporting. And secondly, that reproducibility is not a thing, it's a process. One of the problems that many investigators have is they just don't know what the criteria are. Well, there's the handy tools that we can use, and those are best practice reporting guidelines. Now, the intended role originally was just to promote transparent, complete reporting, regardless of how experiments were actually done. But they can also be used as planning tools because most of these are international consensus guidelines to the minimum best practice standards for reproducibility. And they also outline what the essential tasks are that will enable reproducibility to be accomplished. So specific to preclinical research are two major sets of guidelines I want to briefly touch on today: the Arrive 2.0, Animal Research Reporting of In Vivo Experiments, and the Prepare guidelines. Arrive defines the minimum information required for reliability, validity, and reproducibility. Prepare identifies the key logistic and procedural task areas that are common to all preclinical studies. Now, Arrive 2.0 consists of 21 separate items. We don't have time to go into that. I want to emphasize just 3 today: study design, animal numbers, and methods of bias minimization. So study design is the whole formal, logical, and statistical structuring of your input variables and your controls. Now, formal designs are recommended because they increase the power while reducing animal numbers. And best of all, a lot of designs have been available for at least a century. Most of them were invented in the 1920s and 30s by Ronald Fisher, who invented most of our statistical inference tests. They're still used today. But most studies have no formal design at all. They're either trial and error, or they're just these sprawling, incoherent masses of different comparisons. And there's several reasons for this. First of all, at least in the US, experimental design is not taught at all in most introductory statistics service courses. A lot of investigators, on the basis of one or more intro stats courses, think it's so easy, anyone can do it because you have software, without realizing what goes into the inference tests. And many consulting statisticians, on the other hand, have never done an experiment in their life. And with an overemphasis of mathematics at the expense of practical application, they have really no idea of what's involved in proper data collection and the structuring of an experiment. So sample size is the second essential, and this is the number one reproducibility item. And even more in animal-based research is the concept of the 3 R's: replace, reduce, and refine, so we can obtain minimum harm for maximum scientific value. So numbers have to be justified not just statistically, but also operationally and ethically. And fundamental to this is the definition of an experimental unit, because that's what sample size are. But these concepts and procedures are often misunderstood and misapplied. So what an experimental unit is the entity to which an experimental control intervention can be applied. Say in this case, we have two treatments, A versus B. And in this scenario here on the left, mice within a cage receive one or the other. Well, in this case, the mouse fits the definition of an EU, sample size is the number of mice. However, in this scenario on the right, which is the most common setup in most facilities, all 4 animals in each cage get either that treatment, A or B. This means that the experimental unit is no longer the mouse, it's the cage. So the sample size is not 8, but it's only 2. And this is one of the major causes of problems with the ensuing statistical analysis, is this confusion of experimental units with sample size. Number 3 is bias minimization methods. Now, these are essentially logistic. Randomization, again, is commonly misunderstood by investigators. They think it means haphazard, ad hoc, unplanned, or alternating A versus B. But what it is a formal technical process which assigns interventions to the experimental units with a given probability. What you see in most textbooks is that the purpose is to remove systematic bias from your results, and it does that. But what people don't understand is most inferential tests are predicated on the assumption that randomization has occurred. If you analyze data which has not been randomized, inferential tests will probably be not valid. And then blinding is another logistic procedure which attempts to minimize bias in the assessment or allocation or interpretation of the results by concealing which treatment was received by which subject. And again, rarely correctly performed or reported. So the third stage is building in quality and reproducibility by planning. The poor logistics are the greatest source of study failure, not only because of management issues, which means the study just doesn't get done or completed, but also because of unwanted variation will completely mask any true signals in the results. So the solution is having rigorous study-specific plans, standardized tasks, and standardized training of all personnel in these tasks. So planning has to be study-specific. It's focused on 3 items. What is the research question or substantive problem to be solved? What data will you need to answer that question, and how are you going to get it? And this is where the prepare checklist comes in, because it identifies the 4 major task areas: study formulation, institutional dialogues, quality control methods, and how the study is executed and wrapped up. So in study formulation, this is where, again, another plug for research librarians as well as biostatisticians and ethical oversight committees, because this is where you can actually properly formulate the research question in a form so it can be answered. Literature searches provide research gap analysis, identify best practices. Is the model suitable? Is the research even allowed? It allows you to you have to focus on legal, ethical, health and safety issues, and experimental design statistical analysis. From that, you can also identify the data you need to answer the question by the variables which will have to be measured and how they are to be prioritized. And the third stage, which is almost always overlooked, is how are you going to get the data? Data collection requires logistics and a lot of planning. Who's going to do the work? Personnel, time. Do you have enough money? Do you have space, equipment? Do you have all the resources that you need? And there's a whole group of people here on the right and facilities which will enable you to meet those objectives. So in conclusion, preproducibility, yes, is essential, but it's more than just reporting what has been done. You have to start focusing on building quality into the protocol before the study even begins. And rigorous, robust, and study-specific planning requires validated tools. And this is where the reporting guidelines can really come into their own. So just as a final wrap-up, business as usual is not acceptable. Best practice standards have been evolving now fairly steadily over the last 5 years, and academia has really been a lot more relaxed about this lack of reproducibility than industry has. So you might want to remind investigators that ignoring best practice standards is both negligent and unethical, especially when you're using living animals to perform experiments with. So that's it. Questions, if anyone?
So much, Penny. We got a questions for you. So the first question is, do you see a role for persistent identifiers? And I'm gonna see if you can mute yourself while I'm asking the question, and then unmute. If you see a role for persistent identifiers and data aggregation around the materials for research resources like mice, and the question asker notes that for cell lines, there are some, of course, that are contaminated, but they're continuing to be used. So essentially, what do you think about persistent IDs for mice?
Actually there's RIDs that are specific to strains and vendors. This is very poorly reported. A colleague of mine has just finished a survey of over 300 journals, just focused on oncology, and these are very poorly reported. For example, the C57 black 6 mouse. There's about 40 different flavors, virtually. There's slight strain differentiations. There's vendor-to-vendor variability, and there's a lot of genetic drift. So yes, the better journals are requiring this, investigators are not very good at reporting it, and peer reviewers don't seem to care to pick it up. So yes, it's absolutely essential.
Great, thank you very much.
Thank you very much.
The second question is perhaps a little bit more complicated, and maybe will hopefully familiarize some people in the audience with reporting guidelines a little bit more. And that question is, has arrived with the reporting guideline that you had mentioned earlier, Consort, RRID, and STAR methods guidelines, have they been sufficiently subsumed by MDAR, which is the material reporting checklist that many of journals like Nature and so on have?
No, they haven't. They're very specific to certain kinds of research. An animal-based study has different reporting requirements than, say, a human-based clinical study, which is, or randomized controlled trial, which Consort covers. For example, patient demographics, for example. You don't have to know the temperature that they keep their house in for human beings, but you certainly need to know the temperature and photoperiod for mouse colonies, because that is a major determinant of physiological response, things like that. So no, they're not subsumed. MDAR is general for that journal, but Arrive, Consort are best practice reporting for those kinds of research. And just another plug, Equator Network has the reporting standards for well over 600 different kinds of research. So if investigators don't know how to, say, report a genomic study, they can find all the information they need in one of the relevant guidelines.